Good morning, everyone. Let's settle down. This is the session on diversity and equity. My name is Patricia. I will be conducting this session. We have 90 minutes and 6 presenters, so there will be, it'll be like 15 minutes for each slot. But I would request the presenters to allow some questions, and therefore it'll be something like 12 plus 3. And if a presenter eats up all the time, then there will be no questions for that particular slot. The first talk is actually going to be a set of recommendations. There's a genome diversity network that was formed, and this particular network has met for many times with different groups of people, and they have come up with a set of recommendations. The set of recommendations will be presented by Sophia George from the University of Miami, and Shyam Prabhakar from Genome Institute of Singapore, and Kian from Genome Institute of Singapore. So without further ado, let me invite Sophia. She's going to make the first presentation. Sophia.
So good morning, everybody. So we are going to go over some of the work that we've done for the last year and a half. So why are we here, and particularly looking at this graph, is because over time, there's been an epidemiologic transition from communicable to non-communicable diseases. And you can see that there's variation globally in terms of the diseases that impact different populations geographically. And eventually, of course, those people have different diverse, genetic diverse backgrounds. And what we also know is historically and still, there is a lack of genetic diversity in our data sets. And you can see data from last year in terms of the number of cells and where the people are from in terms of self-identification. And then currently, when you look at the number of cells that are present, what we know and what we don't know. So there's a large gap in what we know, and that information is important for us to be able to do good biology, good science. So as a result of these gaps and these challenges and opportunities to do better, we're motivated with the Genetic Diversity Network to understand both molecular, cellular, and tissue traits that vary across human populations, as I've shown, and that there's these differences in disease risk and pathophysiology, pharmacogenetics across people and populations. And of course, we know in terms of precision medicine, precision treatments, precision prevention, and that diversity is still the scientific frontier. So our goals in terms of expanding diversity, we needed a concerted effort to be able to move the needle forward. And therefore, we decided collectively through the GDN that we needed policies in place to be able to move that needle forward. So we asked how can we formulate these policies, develop goals and guidelines to diversify the Human Cell Atlas.
Great, thank you, Sophia. So the Human Cell Atlas Diversity Task Force was set up by the Equity Working Group to explore these concerns and to advance human diversity for the Human Cell Atlas and also for the genomics community more broadly. So we had the co-chairs, Shyam, Sophia, and I, we're very grateful for all of the members who are listed over here. Samira, Robert, Gideon, Youssef, Vinicius, Ponpan, Drew, Alicia, Yuki, and Patricia. And also the observers, Partha, Musa, and Alex, co-chairs of the HCA Equity Working Group, Jason, Kondwani, Mayumi, and Norbert, who's over there, for their feedback and advice. And also from the community at large, so including the various co-chairs of different working groups and different bio-networks within the Human Cell Atlas, the Human Cell Atlas data wranglers, and also partners within the scientific community. And we were very ably supported, and none of this work would have been possible without the Human Cell Atlas Executive Office, including John, Christine, Esther, Lucia, and Tracy. So what I'm going to do is I'm going to use the next couple of slides to briefly refresh and remind the audience of some of the goals and guidelines that we've developed as a task force in consultation with Human Cell Atlas working groups and the community. So in terms of goals for human diversity, we've looked at multiple dimensions of human diversity because all these aspects have an impact on the molecular, cellular, and physiological traits So for example, we aim for all atlases to have sex balance, so a minimum of 40% male, 40% females per reference atlas. We're thinking about geographical diversity, and I'll have a slide on this later. And we're also thinking about aspects of environmental diversity. So in the long run, being able to incorporate donors and participants from both urban and rural environments. So fundamentally, for all human cell atlas reference atlases, which you heard from, from Gary, from Chloe, and from Ed yesterday in yesterday's talks, we aim for version-to-version improvements in environmental representation as well as geographical diversity. And we recognize that for many communities globally, you can't just have a healthy reference atlas, and to bring about maximum benefit to the community, we really need to expand these reference atlases and include disease and prioritize diseases that are of relevance both globally as well as locally. So here we've got a schematic of some of the human cell atlas goals. You heard yesterday that there are 104 countries and represented in human cell atlas with more than 4,000 members, or around 4,000 members. So in the near run, for some of the more easily profiled tissues such as blood and nasal swabs and skin, we hope to be able to attain increase in representation to at least 50 member countries, which is half of the total number of countries represented in the HCA membership. And in the long term, so in the next 10 years, at least half of BioNetwork should reach the same 50 country goal. And ideally, in the long run, we want to have all 18 BioNetworks that you heard from attain this goal. So we recognize that this is going to be challenging, especially for tissues that are more difficult to profile. I'm going to end with this slide on the diversity metadata, and I'll encourage you to actually reach out to the Human Cell Atlas data wranglers who have a poster at poster number 42, and they've also included the human diversity metadata within their schema over here. So we encourage and recommend that all teams contributing to the Human Cell Atlas reference networks, to reference atlases, including including your contribution of tier 1 integration technical metadata, as well as tier 3 cell annotation metadata via the cell annotation platform that Chloe so nicely demonstrated yesterday. We encourage all teams to think about what kind of donor metadata and aspects of human diversity you can capture. So all of the fields that are in black colored font over here are some of the core fields that we really recommend all teams collect. And we'd encourage teams to also think about some of the ones in blue that are useful for thinking about various aspects of human diversity. And before I hand over to Shyam, I just wanted to mention that we're delighted to announce that the Diversity Task Force report has been ratified by the Human Cell Atlas Organizing Committee. So we're glad to be able to push forward and be able to drive the implementation of this. So thank you to for the ratification to the HCA Organizing Committee. All right, Shyam, over to you.
Thanks, Kian, Sophia. Thanks to everyone in the community who contributed to this task force deliberations and decisions. I hope this can guide, provide a firm footing for how we go forward in the Human Cell Atlas to incorporate diversity. But beyond, I would say, beyond our terms of reference, there was one major additional contribution that the Diversity Task Force made, and that is something that came from the community. There's strong feedback across the board saying that to achieve diversity, you have to tackle disease and show benefit to the community. Without that, it's hard to recruit participants in many parts of the world. It's hard to get the local funding bodies, local researchers enthused. And so very glad to say that that message has been taken on board by the Human Cell Atlas. And it is part of our roadmap, as Aviv and Sarah and Chloe and others showed yesterday, to tackle disease as well in the Human Cell Atlas and develop a scientific basis for diagnostic criteria for therapeutics management, therapeutics development, and so on, and preventive care, of course. So how do we decide which disease to tackle? We cannot tackle all diseases, it's an ocean. And really we have to play to our core strengths in the Human Cell Atlas, which is healthy reference atlases, but of course as a means to understanding disease. So we want to do case control. A handful of diseases that are prioritized based on global and local disease burden, rare or neglected diseases, and then of course, what resources do we have, what expertise we have, what interests do we have in the Human Cell Atlas community, and then how are we complementary or synergizing with other consortia. We don't want to reinvent the wheel. So based on these criteria, we reached out to the HCA community to say what are your priorities, what are you interested in disease-wise. And pleased to say that over 200 responses were collected, so that's massive, across the bionetworks. So you can see here a map of the, how the bionetworks broke down in terms of number of samples. Seems like breast and gut had the largest number of samples. In terms of number of studies, perhaps brain had the largest number of proposed studies, then colon, kidney, skin, breast, and a few others. So this gives us a strong basis for selecting the subset of diseases and conditions that we will tackle in the Human Cell Atlas. This won't be the only criterion, of course. It depends on funder priorities and so on as well. But this tells us what we can do. In terms of specific disease areas, metabolic disease, not too surprising, so metabolic disease, cancer came up. And then since immune is such a strong component of Human Cell Atlas work, autoimmune and infectious disease came up as well. You might have heard these disease categories before in various Human Cell Atlas global meetings and regional meetings, right? So now we know what to focus on, and we can narrow it down even further. Coming to some actual science, we talked about how there isn't enough diversity in global genomics, but there are a handful of studies now coming from the Human Cell Atlas. So this is one where we looked at diversity in Asia and found that sex, age, and ethnicity had strong enrichments in specific cell states in human blood. But this is all healthy. Moving to disease, this is a study from Sophia's team looking at high-grade serous ovarian cancer, showing the violent plot shows that Nigerian cancer versus cancer in the US has systematic differences in cell proportions. And maybe this can help to untangle why disease incidence, disease severity, disease outcome, and so on are different in different parts of the world. So these are just examples of exciting work that's happening in the Human Cell Atlas. But hope you will all join us in this effort based on the recommendation of the Diversity Task Force, based on these principles. Contribute your data sets, and not just data, but contribute metadata as well. Brainstorm with us, help us organize the community, organize funders to tackle these diseases. Thank you.
Thank you very much, Sophia, Kian Hong, and Shyam. I'm pretty sure that a lot of you will have comments, questions to ask, but let's stick to 2 comments or questions. Yes. Yeah, Christine.
I wanted to say thank you and congratulations on a really fantastic report. It's very comprehensive, and I'm really proud of it, and I'm not an author on it. So one thing I wanted to highlight in it is that it's not just motivating why we should be building diversity into our atlases, but also making very strong statements that the way we do it matters. And I would really love to see more discussion on that aspect too. Thank you.
Thanks very much, Christine. As a matter of fact, we didn't dwell on it, but the Diversity Task Force recommendations goes into some detail on how we go about this to maximize benefit and minimize harm. And thank you for your active participation in that process and all your suggestions.
Anyone else?
Thanks for the hard work you all have done. This may be a political question because in US now the word diversity is not the word we are allowed to use in many ways. So I know many of us are involved in this research. I'm just wondering how can we tackle that issue moving forward at the global scale?
I think our 4th speaker will take this question.
I feel sort of uniquely positioned as someone who had to find synonyms. It doesn't change at all our mission. We're just going to use a word, for example, global representation. And so we're not changing our objective. We're not changing our mission. There's different way of explaining the impact of diversity, and global representation is somehow palatable. And so no worries. We're not changing the mission.
And just to add that this is why UNESCO is on board.
We're so grateful for UNESCO. Yes, we are backing.
Thank you, Chloe. Thank you, Casimiro.
Thank you, Chloe, for that. All right, so we'll move on to the next presentation. And again, with the hope, first of all, let me thank all of these people for working so hard for over a long period of time to get to these set of recommendations. Our next speaker is Shin, who actually works on single cell and spatial and looks at non-coding DNA and RNA and so on and so forth in various diseases. Today, he's going to talk to us about mapping human diversity using spatial. Jay.
Thank you, Partha, and good morning, everyone. I want to briefly introduce some of our ongoing efforts to how we can expand the Human Cell Atlas Asia chapter. But before that, I want to give you a quick overview of the regional representation here in Asia, where home to 60% of the world reside here in this part of the world, and it's most populous countries such as China and India are in Asia. And therefore, just naturally, there are abundant genetic, ethnic, cultural, environmental, socioeconomic diversity. However, it lacks central governing body. So how can we work together to bring the scientists and researchers and all the clinicians and participants together? And as you just have seen in previous presentation, there is a strong impetus to really create more diverse atlas and have a more representative and equitable data sets. So therefore, we kind of formalize our efforts to create through a creation of steering committee. And you see myself, Shyam, Unyang, Yuki, Pampan, Sejunti, and Shogang. And along with Piero, Ando, Yoshi, and Esther are also helping us to really bring this energy and synergy to recognize the importance of genetic diversity and representation in the region. And so we are really thinking about what are the key missions that we need to implement in order to drive this equitable and diversity to come to realization. So first and foremost, we do want to build key community and bring people together by strengthening regional research alliance, really, and also have an opportunity to showcase what are the latest and coolest things that are happening in the region, and therefore identify key synergies and promoting such collaborations. And as a result, we hope to then diversify the Human Cell Atlas. So we are working very closely with global HCA, and then we also coordinate annual meetings and workshops, along with working with the Diversity Task Force Equity Working Group. And we also provide key communication and ethics toolkits to also enable various countries in the region to reach the level of diversity and equitable science. So we do go through annual meetings and we identify key flagship projects, and they usually revolve around disease, cancer, infectious disease, aging, gut biology. We're not limited to those. We're open to other ideas. But really the idea is to bring people together with common interests and seek funds together, and perhaps we can support some of these aspects and provide central data portal to really achieve the ultimate goal, which is to get the equity and diversity and to have more meeting the local societal and disease burdens, right? And so I think just formation of the active members who are really aligned with our interests have been extremely helpful. And we were able to organize multiple meetings all around Asia. The very first meeting was in 2017, co-organized with Piero and myself in Okinawa, Japan. It was a very small group, but it was a really good starting point to really recognize the importance to have this HCA Asia chapter. And in 2018, actually in Seoul, in Jeju Island in Korea, organized by Unyang Park, was the first time when Unyang, myself, and Shyam got together and said, hey, we should really do something. And that's really where the whole Asian Immune Diversity Atlas discussion came about. So the whole point is, I think meetings like this is a perfect venue to really interact with people around you and think about what are some key research interests that you guys are able to collaborate on and really engage with key stakeholders and funders to really pitch your ideas. And that's literally what happened in 2017. And as a result, we were able to successfully complete the map of the Asian Immune Diversity Atlas that you just heard as well. So again, it's a collective effort across multiple countries in Asia, and perhaps it's the most diverse immune atlas to this date. And it's across 1.2 million PBMCs, over 619 individuals. And again, led by Kian Hong on this paper, was a really testament that we can really build the community together and make it really impactful for HCA. I won't go into much detail, but there are definitely, we do discover key ethnic differences and gender differences, and in fact age differences throughout PBMC proportions. So it's really important to understand the variability of healthy reference. And so I think it really calls for efforts to expand on this collaboration in the region. In fact, the data set is so rich and very resourceful. There are multiple papers that also spun out of that consortium data set. And Bo Xiang had a wonderful story about splicing QTLs based on IDA data set. Yuki's group have talked about X chromosome inactivation based on IDA data set. And Jo-Eun Park from Korea is looking at TCR repertoires and how clonotypes of TCR varies across age using that data set. Together with Hyo-jae Kim. And Chung and myself are looking at, can we enable long reads as well from this PBMC data set, and looking at also regulatory elements and mapping enhancer RNAs from these IDA data sets. So it's amazing resource, and hopefully many of you can have access to, you guys have access to this, but just again, a testament that a good data set can really expand and have much broader scientific impact than what you initially intended to do. And really, that's really the heart of the HCA effort. But, as we discussed about, for the past several years, what's after IDA and what can we do beyond immune cells? And here is a snapshot of the survey of the HCA spatial survey that has been going around. And here's just a general distribution of various tissues that are being profiled and being conducted by the various groups. And here I just want to point to skin. And you'll be asking why skin, why is it important? And it's not because I'm Korean, by the way. I think skin is definitely important, and I'll explain to you why. In fact, it is the largest organ, so it is most accessible and it's very relatively straightforward to sample. And because you're just biopsying from the skin, you can actually do longitudinal studies where you can actually understand how age can impact the skin biology. So it's a very nice model in a way to understand how cell interactions can change over your age time. In fact, it's also very complex. You'll be surprised the amazing interplay between epithelials and mesenchymals and immune cells and their interactions with scalp and hair follicles, vasculature and sweat glands and adipocytes. So it is a very nice and well-demarcated structure that we can understand the interplay of the cell types. And there are really well-established 3D models that you can really also consider perturbations or genetic manipulations in some of these patient-derived skin. Not only relevance in longevity and aging, also not just cosmetics. Of course, this is a huge interest in industry and probably many of you in the crowd, but also disease are quite prevalent, especially in the region where atopic dermatitis, psoriasis, and other allergic reactions can also have huge burden in society. What's also very relevant and interesting is environmental and geographical factors, such in Asia, ranges quite a bit. You have tropics here in Singapore, it's humid, but maybe in the northern part of China, it's very dry. And these could have very different predispositions to certain disease or onset of skin pathology. And UV exposure and pollution, such as exposomes and metagenomics, bacteria that are growing on your skin, can also have huge impact on the biology of the skin. And so I think collectively, I think skin really is a really interesting model for us to study. And so we've been surveying more locally for the past few months about what are some of the skins that are available in Asia. And here's just a rough estimate of some of the samples that have been reported along some of the friends and colleagues that we know. So, collectively, we can estimate about 494 FFPE sections or blocks from healthy individuals. And some of them can have access to autopsy samples or postmortem or cadavers, which would allow us to sample tissues or the skin from multiple regions of the body. And when we talk about disease, we have roughly 343, and I think these can also increase as we engage more with the community about clinical samples. So it's not just Asia, but beyond Asia, there are Australia, America, Europe, Africa. With Muzz, we've been talking about how to build this atlas together with European counterparts. So I think this will be a great opportunity for us to tap into the latest spatial technologies to understand genetic diversity as well as the skin biology. So to test this, we've been trying to understand how we can leverage spatial technology more on the skin. Here's just an example of how we are using spatial technology to understand different skin biology. This is an example of a alopecia areata as part of baldness that is caused in your hair scalp. But with contact immunotherapy, it actually regenerates the hair growth, induces hair regeneration. So we wanted to understand what are the immune response during this contact immunotherapy. And this is just a quick overview of applying VZM HD into some of these tissue sections. What you see on the left-hand side is the non-responders. So even if the contact immunotherapy, they don't regenerate hair. And on the right-hand side, you have successful regeneration of the hair, and you have very distinct clustering that are observed in the skin. And you can annotate them and appreciate the fact that, in fact, the responders have higher immune cell recruitment towards the epidermis and dermis layer, whereas the dermal fibroblasts, relatively, the quantity stays relatively the same, but then the dispersion of the fibroblasts are quite different in the responders versus non-responders. Just want to caveat that their fine-grained cell annotation still remains a challenge, and we are trying to explore how we can better improve the annotation of subtypes of immune clusters, and we'd love to discuss with the whole community here of how we can really improve this resolution. But skin being skin has a very distinct layers of epidermis, and we are able to cluster them very nicely with this technology. And in fact, every layer can have very distinct differential gene expression patterns. And because it's a whole transcriptome data, we're then able to understand the gene set pathways and what are some of the contact immunotherapy induced and differentially expressed genes for individual layers of the skin. And although we are not able to pinpoint individual immune cell types, we are able to overlay some of the key marker genes, and we can appreciate a key accumulation of marker genes that represents T cells and a collection of chemokines. And you're able to see a co-clustering of these cell type and the ligands in the responder patients, but not in non-responders. I want to switch gears a little bit and just show you some of our current data on Xenium, and this is normal skin, and we were able to perform 5K pan tissue panels, and here you can also appreciate different representation of the epidermis and expression of specific chemokines, as well as a subtype of CLS12A2 genes in different parts of the human tissue. And I think you can appreciate the granularity and their segmentation of individual cell types in the skin, and you're able to overlay some of the gene expressions on top. So what's really nice here is then you can really zoom in more, and again, try to elucidate what are some of the cell types that are represented and what are the ligands and chemokines that are secreted. And again, you're able to understand clusters of CCL17s in certain spots near the endothelial cells and the recruitment of certain immune cell types, such as T cells and macrophages in the vicinity. So these are granularity that we're able to see with the Xenium, where it was very difficult to see this with the VZM HD technology. So again, we've been yesterday at the technology discussion, what are the pros and cons of spatial whole transcriptome versus imaging data. I think there are no right answers, but again, it really depends on your question. But HD gave us a large number of differentially expressed genes to understand broad pathway analysis, but here we're seeing more cell-to-cell interactions that are quite relevant. Here's just another example of two different age group, where 29-year-old female and 53-year-old female. And we do see difference in clusters of chemokines, again, suggesting that these could potentially be some of the aging markers and some of the immune recruitments that are happening in the process. This is not really a skin tissue, but our colleague in GIS, Minghao Chia, has a way to also visualize microbacteria here on the surface of near the colon here in this example. But we are thinking about how we can apply this also in the skin and understand how different microbacterial culture can impact the biology of the skin and transcriptome. So I'll just end here by saying, I think hopefully we're building upon the success of the IDA project. We're really envisioning a strong regional effort to create a comprehensive Asian skin atlas. And we hope to really uncover diversity of the skin across ethnicity, geography, and environment, so that we can link it to aging and disease and environmental exposures. And again, I think skin is a great model to study aging through ability to biopsy over lifetime. And also, hopefully, we can deepen our understanding of human biology and precision dermatology and make it more translational. So I said this a lot as one person standing here, but I know it cannot be done by myself or a single group or a single country alone. So we do require you guys and the whole community to come together and also work closely with the Skin Bio Network and dermatologists, clinicians, and researchers and technology partners to make this happen. So with that, I first want to thank the whole IDA group who really spearheaded the whole effort to bring community together. And this is just a picture from our Hong Kong meeting last year. And also want to thank other members of Spatial team, Aging Skin, and alopecia areata. And yeah, and thank my lab members. And happy to take any questions.
Thank you, Jay. We have time for just one question.
This is superb, Jay. Also, I love the vision. You're appealing to the geeky immunology side of me. The responder all had very high interferon-stimulated genes. Do you know if it was more driven by the fibroblasts? Because when you were showing us responders and like layer
Correct.
4 and 5, you had all 6, 9, 10, 11. I mean, usually monocytes, macrophage, may produce it, but so are fibroblasts.
So what we see is that the responders have these are actually contact immunotherapy that activates Langerhans cells, which reside in the epidermis. And then they recruit the monocytes and dendritic cells, and they further activate the T cell recruitment. So these are, by definition, alopecia areata it's an autoimmune disease, and the T cells are attacking the hair follicles. But then by definition, by contact distraction, you're trying to bring the immune cells to the surface of the epidermis, away from the hair follicle, so that hair follicles have chance to regrow. And that's actually what we're seeing, is that immune cells are recruited to the surface, away from the hair follicles, because Langerhans cells are recruiting them towards the epidermis, if that makes sense.
Cool.
It's very cool, yeah.
And just congrats, this is a great vision also, just overall.
Yes.
I hope that more bio networks and effort will follow your way. Well done.
Yes, I think fibroblasts will definitely play a role. We just haven't explored that angle yet.
Thank you, Jay. Our next speaker is Patricia Severino. She is a group leader at the Albert Einstein Research and Education Institute in Brazil. She works on cancers, and essentially the question she tries to address is, why is there so much difference in immune response in cancer, individuals with cancer? She's going to talk to us about why diversity matters. Patty.
That's it? Is it? Sorry. Is this on? Yeah?
Yeah.
Can you hear me?
Oh, yeah.
Cool. For this talk, I decided to invite a colleague that we discuss a lot together about diversity and Latin America, Yesid Cuesta. He's from Colombia, and he's probably listening to us, so we put this together. And we decided to talk briefly about how important it is to expand discovery through genomics, but also researcher inclusion. So why are we here? We're here because the Human Cell Atlas aims to map all cell types. And why does diversity matters in this context? So without global inclusion, the Human Cell Atlas would be incomplete. And there would be gaps in discoveries and inequitable healthcare insights, which is what we want to avoid. Why then regional genomic diversity matters? Why diverse ancestries and regions? So in terms of ancestries, the genetic ancestry influences immune responses, metabolism, disease risk, and cellular behavior. Yesterday we talked a lot about this during the flagship projects discussions breakout session, and also at the equity breakout session. So those who were there probably heard a lot about this, but not the others, so I thought it was important to bring it. And when we talk about regions, environmental exposures and historical events also shape biology. So all that would be missing if we do not include this diversity. So scientific quality depends on diversity, and then we're not talking about just representation of populations, but also biological comprehensiveness. I brought here a personal example about the importance of genomic diversity and why it matters. Back in 2019, I was collaborating with Musa in one of his very innovative and creative projects. He has many of those. And in this project, we were studying, or his idea was to study a long no-coding RNA, a specific variant, genomic variant, and how all that would influence inflammation regulation. And I was interested in that because I work with sepsis, and we were trying to understand how some patients would recover from sepsis and others would move on to septic shock. And that was all related to inflammation. And at the time, we didn't know how the impact or the impact of that specific genomic variant in the Brazilian population. And just most recently, I brought here a paper that is down here, was just published last month about the diversity, genomic diversity in Brazil. And they have almost 3,000 genomes that have been sequenced. So now we can actually really talk about diversity and genomic diversity in Brazil and understand this impact. And with Yesid Cuesta, we work with genomic diversity of bacteria in hospital-acquired infections and how hosts respond to these infections. So we also need to understand the genomic variation of the population to be able to link those two diversity matters. And how can we then make sure that HCA does not follow the steps of previous projects? So a lot, all we know nowadays is based on very little diversity, very narrow subset of global populations. And we are working towards not falling into this same situation as we know it. So we believe that we are talking beyond genomes, beyond the inclusion of populations, but the inclusion of regional researchers too. So local scientists, they bring deep knowledge on local health priorities. They have access to, and they also have trust with diverse populations locally. And they bring a contextual interpretation of biological findings that otherwise you cannot have without including these researchers. So we need, obviously, capacity building for this to happen. And I would like to showcase the model by CZI, who funded ancestry networks. For us, this was a very fundamental opportunity across Africa, Asia, and Latin America. Without this kind of funding, we're not going to be able to do what we're going to show here, or what Jay just showed. You know, like, it's amazing. We like follow the footsteps of Ida and Asia ancestry network. So the idea of this funding was obviously to generate high quality ancestry diverse single cell data, but also to build local research capacity. So funding diverse research teams, not diverse in the sense of many, but diverse in the sense of really the teams, you know, that what they brought to the projects. So we have projects which are led regionally, so data generation and leadership locally. So not just participants in the research, as included recruited people, participants, but leaders also in discovery locally, and that's very important for us. I'm showcasing two projects, the one that I take part in, which is Latin cells. Here you can see the map of Latin America in the middle and how we are sampling and including populations from very, very diverse regions. Here I show the diversity of local researchers. So all of them are based in Latin America. They share different expertise. The expertises, they are complementary, and this is very important to us. This is how we intend to, next year, bring data from our projects to present to you guys. And our cousin project, Jaguar, from Yesid Cuesta is part of this team, and it's also in a very similar way joining different researchers from different countries in Latin America. Very similarly, it's led by Gosha in the UK, but it also brings together the opportunity of sampling and building local capacity in Latin America in a very similar way to what we're doing. So we talk a lot, and the intention is that we are able to put all our findings together in the end of the project. So there are challenges but also solutions for us to make this happen. So how can we include more of local research and researchers? So barriers are mostly related to funding disparities in the countries, and that's why I really wanted to highlight the contribution and the vision of CZI that funded the ancestry networks. Infrastructure gaps that sometimes we have no idea how big they are. We sometimes underestimate the gaps that we have, considering the different countries. And there are a lot of talks always about data sharing and data ownership. So it's very important that partnerships are very transparent from the beginning, that everybody is aware of how things are going to be done. Training, so capacity building, which we are able to do now, thanks to this very visionary kind of funding model that helped us, and obviously also equitable data governance. That's something that is also very important. So finally, for a truly global human cell atlas, we must expand not only samples, but also our scientists. And for everybody who is here that can help with that through advocating for funding, diversity, build equitable collaborations, and to make it a priority to have regional inclusion in project designs. So this is what I wanted to show you. And we have a very simple slide for thanking the whole Human Cell Atlas community, because since the beginning, they've been really engaged with the idea of inclusion, not only the equity working group or ethics working group, but everybody. The scientists, they have always been present to share what they know and what they have discovered. Since the first meeting that we had back in 2019, they were really eager to participate and to help us out. And CZI, obviously, like I said, without this kind of funding, we cannot make it happen. And our groups, so represented here some images of our team. Not everybody's there. Most students are not there, but it's a very big group, and a lot of integration is going on these days. So, and finally, there is going to be a Latin American session on the June 12th. So you want to see a little bit of what's being done in terms of single cell research in Latin America. Please join our session.
We have time for a couple of questions or comments.
Hi, Patricia. It's a wonderful presentation. I have a question regarding the indigenous Americans. What kind of efforts are you making? Because with the limited data we had in the breast samples, we could see at a single cell level a different subpopulation of cells. But I'm a bit cautious with our data because the number of samples were limited. And based on the data you presented in Brazil, there is a portion of the population which are untouched. They are pretty much pure indigenous people. So are you making any special efforts to collect those samples for any of your diseases?
Yeah, for indigenous populations, that's been mostly carried out in Peru, Colombia, and mostly Peru and Colombia and Mexico. In Brazil, we're not collecting samples from indigenous populations which are isolated. We just collect from indigenous populations that are urban, so the ones that are living in cities. I don't know if this answers your questions, but yes, there is a lot of different care and ethics and the way to deal with these populations. Like I mentioned before, you need researchers that are like trusted by these communities and have been engaged in with these communities for the longest time, you know, so it's about that. You cannot just approach them for a project like that. So sample collection is based on years of engagement with these communities.
If I can add just one comment to what Patty just said, also these isolated populations, even though ethnically they are very interesting, for single cell research it's very difficult to get samples from them and bring it to the lab. We can't take the whole single cell lab to the to the field, so it becomes very difficult. So like I said yesterday in one of the sessions, we need some kind of a practical approach, scientific and practical combination. So that it will be a challenge. It will probably eventually be met, but not right immediately. There's another question.
I'm just going to make a comment about that, because in our Latin Cells project, we do take the lab to the communities. We showed it last year. It's really complicated. We have amazing pictures if you guys want to see it, but the whole lab does go to the communities, special cars, and we did use a protocol in the whole team that does not need refrigerators or cold centrifuges, because otherwise we wouldn't be able to collect samples in the field. But it's a very good point. It's very complicated. So we were relying exactly on people who had been doing this for the longest time in Mexico, Peru, and Colombia.
One last question there.
Hi. Also, just want to mention that thank you for your slides. And I don't know much about Brazil, but I know that Brazil is a very diverse country from my friends from Brazil when I studied in the UK. And I also saw Daniela Robles Espinosa, who's also one of those who were helpful during my doctoral studies. My question is, part of the concern I have when I was doing a study is the normalization of ethnicity-specific samples. So particularly I studied the British cohort on primary melanoma patients. And I learned that because we don't have much normal samples, so I had to filter out samples from the 1,000 Genome Projects, only specifying how do you call this, unique set of people of certain origin to correct the samples from wide population. In Brazil, knowing that through the effect of migration, there are Brazilian Japanese, and I know also that a lot of Brazilians can have British passport, German passport, and all of passports from Europe that gives you so much access to the world. And how do you correct for those very tricky form of diversity in terms of your studies?
Well, as a matter of fact, this is the probably one of the main objectives of the project, which is to access this ancestry background. So we are doing genotype and genome sequencing to understand this diversity, and our results are going to be based exactly on the ancestry background and on this mixture. So we are mostly collecting participants in São Paulo with all the background from the rest of the country. So we have all this diverse ancestry, but probed in São Paulo due to this environmental regional impacts. So we believe that we are going to be keeping some of these effects away, considering the region, and then just incorporate the ancestry background in the analysis, like you're saying. It's very diverse, but that is the point, to understand the mixture. Contrary to the other groups that are studying very more isolated populations, so their conclusions are going to be on specific ancestries, and ours is going to be on the diversity.
Just a question. How far in history can you go? Because when you talk about Hispanic people, but they were already mixed when they arrived in the American continent. They were Arabic, Romans, Vikings. So how far can you go to see the difference? Because I mean, Europeans are mixed by themselves, and when they arrived in the 16th century, there was already a mixed population.
I think if you read this paper that I mentioned on the diversity of the Brazilian population that was just recently published, they do talk a lot about this mixture and how far it goes. I think it would be, we can talk also about it later a bit.
Thank you, Patty. I think we should move on. Our next speaker is Christian Happi. Thank you very much. Christian Happi is the director of the Africa Center of Excellence for Genomics and Infectious Disease in Nigeria. His research focus is on human genomics, molecular biology, and genomics of infectious diseases, especially malaria and viral fevers. Today, he's going to talk to us about genomic surveillance and characterization of microbial threats. Christian.
Well, thank you. Well, I'm going to talk about something a bit different from what you've been hearing in the past, since yesterday. And eventually in the end, I'm going to link it up with the potential for diversity on the continent and then the human-centered atlas. So as you all know, Genomic has really changed the way we are approaching research and have brought a lot of benefit for healthcare and others. But not much of that has really accrued some tangible benefit on the continent of Africa because of many reasons. One, infrastructure, two, human resources, and then many other things. So, and in order not to miss what I call, again, the major 21st century revolution, we really thought about how we could apply this, in the context of Africa. So many years ago, so I think we sat down and then looked at it, and then you think about a continent that is rich in everything, mineral resources, human resources, but also I think a very powerful thing about the continent is actually the microbial biodiversity on the continent that should be considered as a wealth, but not really a challenge. So we set up this platform basically just to address this problem that is basically built on academic and research institution in Africa and then over at Harvard where I used to work. And basically just addressing two things. So one is genomic education, and then the second one using genomic research to address problems that are very, very convergent across the African continent, and then those problems are really based on, it's the fact that most diseases on the continent of Africa, infectious diseases present with fever. So, and then the question often is that when you have, a fever, so what really is driving the fever illness? So I think our project was really to start seeing how we can use microbial metagenomics to have an idea of the microbial landscape, understand what are the drivers, and then eventually also look at the host to see exactly what makes some people sick and then others not sick in the same environment. And in doing this, we're basically addressing one of the most critical needs for education and research in Africa, and that's really genomic capacity building. And in recent, the goal for us was actually to build our next generation of African pathogen hunters in the death rows of Africa, within Africa, and how we leverage, the knowledge and skills, and in order to transform the way we can not only understand pathogen, but also respond to epidemics. Then we set up this platform, and then not quite a few months after we said this, we actually faced our very first, what I call baptism of fire. So we had to respond to a major epidemic of Ebola in West Africa. And that's where we actually realized how innovation can be very impactful. So what we did basically at the time was actually to transform the way we can respond to epidemic. Prior to our using genomics in real time to respond to an outbreak, what used to happen is the fact that the WHO will send experts in quote that will come in Africa, and then they will take the samples to some lab in the US, or in Europe. And then 2 years later, they will tell you, oh, what killed people during the outbreak was this particular virus. But we decided to bring innovation and do these things in real time. So when we had that outbreak in 2014, we actually started doing underground genomic sequencing, pioneering real-time genomic sequencing for outbreak response. We sequenced the first 99 viruses, made the data openly available for the global health community to use. And then most importantly is the fact that we went on to actually advance the protocol and accelerate sequencing on the ground. So a process that we take 3 weeks at a time, we're doing that within 10 days from sample collection to submitting the data in GenBank, and eventually publish our papers virtually a year later on. So basically showing the world how you could do things and do right. But we also discovered, and pioneered the whole idea of discovering variants that are responsible for increased transmissibility during the outbreak. And then you could see that here we identify several mutations, identify one particular mutation that was actually associated with increased virus transmissibility. But most importantly is the fact that for the first time during an outbreak, as you see here within space and time, we're using genomic data actually to show transmission, in people. I mean, it's showing exactly transmission chains and also identifying hotspot, and then show how you could use this kind of data to guide governments and then to show how they can use, to respond to an epidemic. But most importantly, we introduced another concept, which is called translational genomics. We went from genomic sequence data to create the very first rapid diagnostic test that was approved by the WHO and the US FDA under emergency use authorization. So you can see through the narrative that I'm building here that we actually set up the framework that the world used in 2020 to respond to COVID-19. Everything that we did in 2014, real-time genomic sequencing, open data sharing, discovery of variants, translational genomics. If we didn't do this, showing clearly that you can go from genomes to creating a solution, which is diagnostics. People like Moderna wouldn't think about how you could go from genomes to create a diagnostic, create a vaccine. So we did this within months. So I think that's a clear demonstration that this worked. But the most important demonstration of our approach to respond to epidemic is what you see here. This is Nigeria, and this is a church. This is a Friday. I took this picture myself. And then you could see 3 million people gathered in one single spot. And this is 2014 during Ebola outbreak in West Africa. And you can imagine, just looking at this, imagine if you have a few individuals in this crowd that have Ebola, then we'll probably end up with a major, what I call cataclysm. But again, we use this same approach that I showed you to do what people thought was impossible. We use this and we beat Ebola in 93 days in Nigeria with only 20 cases and a death. That shows the power of this approach, for instance, to what we're doing. Then fast forward, for instance, to a virus that many people were neglecting. I call it a virus, Lassa fever. Lassa fever virus is a neglected viruses. But we gave prominence to this virus, not only just sat down and then address it, but again, use the fact that you could sequence this virus and then make it available. And I think 2007, I didn't know much about this virus, but then eventually started looking into it. But then we sequenced the first 1,000 of this virus. A virus that nobody knew anything about before. But again, it enabled us to understand the history, the ancient origin of the virus. We were able to know that this virus emerged from present-day Nigeria 1,181 years ago, was circulating within Nigeria for 600 years, moved out of Nigeria 400 years ago to the West Africa. But most importantly is the fact that from the time where we actually just sequenced this virus, we accelerated vaccine development. So within 5 years of sequencing this virus, we now have a vaccine, on phase 2, going to phase 3 clinical trial in the country. So, and then in addition to that, we have also to develop this rapid diagnostic test So our approach for this is actually being translational. So we're not studying a pathogen because we want to study it. We study it because we want to make sure that we find solutions. So we're not describing it, finding a solution and provide it to the people. So most important is also leveraging technology and innovation. So when CRISPR and Cas and everything became available, we actually adopted that and created the very first CRISPR-Cas, for an infectious pathogen. So, and then that, you could see that in my lab here where we have Amy Maxon that came actually to see it. Very, very powerful technology that makes it very cheaper, affordable, and then also faster. So you could see it, that's the output. But for us, this is where the link, comes in. So you're working in the field, you see people that are exposed to a virus, some die, no matter what you do, and some other ones, you see when you do PCR, you see that the virus load is very high, but you see them ambulatory. They are not sick. So the question is often what makes some people in the same environment sick and others not. So, and then just like the previous speaker mentioned, so we decided to do genome-wide association study to actually see, what is going on in those populations, a case control study. And then we found, a very good marker associated with resistance to this disease. And that explained the reason why you see some people sick and others not. And that you could see that here, chromosome 22 is on the large gene. And we've been able also to explain that biologically how this happens. So alpha-dystroglycan is these putative receptors for all world RNA viruses. It recruits another protein, subprotein called large. And then the large protein, the human genome for many of you know, when it is a wild type, so that actually facilitates easy entry of the virus into the cell. But then when it is mutated, as we showed, for instance, that actually blocks the entry of the virus into the cell. So, and that for us was very important, and we actually do a lot of work right now to actually to see how we can actually make it, turn this to something more important. But again, importantly is also how we leverage this technology not only to do surveillance, but also to make discoveries. So this is how we use metagenomics, for instance, to discover the 2 novel viruses in the southwestern part of Nigeria. But I think what's important here is if you're looking at what I'm saying, you will see here that we actually accidentally stumble into healthy. When you do a case control study, you think you're going to be finding a lot of viruses in people that have fever, but we find this in BS people. And we actually went further to probe, and what we observe is that, if you basically doing this kind of study in our own environment and you are using fever as your denominator, then I think you might be wrong, because in this particular group of people, we found that they were taking all these traditional herbs that that many people take in the context of Africa. And when we recruited them, their temperature was very low. And then when we went back to the communities also to actually show that some of these viruses that we think are new have been circulating for many, many years, as you know. 87% of people in those communities actually were exposed to these 2 viruses. But then again, fast forward to show you how we could use this to do excellent work is show here. This is a case of 179 children dying in southern part of Nigeria within, a week. And there was a lot of panic. This is post-Ebola. We thought Ebola was back in country, but we were able to use real-time metagenomics to characterize, a yellow fever strain that have not been circulating in Nigeria for the past 94 years. That's what you see that clustering right here. Then this information was given in real time to national public health authorities, and then they were able to contain this within 2 weeks, saving thousands of lives. This actually prepared me and my colleague Patricia at the Broad Institute to really start thinking about presenting a novel idea. That was in 2019, an early warning system for pandemic prevention and response. But at the time, we just built on what we've done in the past. And then I didn't know that at the time that in 2020, I was going to face another challenge. So I think some people do tell me that don't come up with new ideas because we'll be having trouble very soon. So, and as it happened, as you see, COVID actually started. And in the case of Africa, for instance, you will see, so Egypt was the first country to report, South Africa followed, and Nigeria was the 3rd. But the difference between Egypt, South Africa, and Nigeria is that for weeks, Egypt and South Africa did not know exactly what they were dealing with. But in the case of Nigeria, within 48 hours, we had the virus fully sequenced, and importantly, we shared the protocols that we had to the whole of the research community in Africa. And you will see our South African colleagues and friends becoming champions, discovering the Omicron variants and others sharing with the rest of the world. That actually shows the power of sharing and sharing data. This basically just giving you a sense of what happened within Nigeria within a few weeks. when you use data, genomic intelligence to inform government. So we know at some point that we have like 49 different variants circulating in different places, giving this data in real time to government, and then they were able to use that to identify hotspot, block, and then do it. But importantly is that actually within weeks, I would say 2-3 months, from our first sequence, you could see here the first CRISPR, as for SARS-CoV-2. And we did this, you could see very raw there, but eventually that's the final thing that we have is, power-free, so saliva, and then you could use that clearly to actually do diagnosis. But then we leveraged this to train a critical mass of young Africans. So over 2,500 and then almost 3,000 across the continent. Africa is 54 countries, but we've been able to reach 53 of the 54 of the countries. But now, we've also learned a lot during that epidemic. You also learned through me that, we also learned that during that time, the question was, why were many Africans not dying? A lot of people will say, oh, they're not testing, oh, they are not doing this. It's a combination of many things. There was the science, there was the knowledge, there was the experience, there was the skill, but then there was also something very important that's probably linked to our DNA. And you can see here, Pablo, for instance, pre-Nobel 2022, looking at ancestral or ancient DNA, actually found something very important, and did find some alleles in those ancient DNA that could be associated with protection, to this disease. So, and that really is going forward is how do the reasons why we should invest in African genomic variations is actually there are 3 major reasons: ancestry, ecology, and equity. So, and then why this? Because, for instance, we've shown that if you take very few samples from Africa and then go on and sequence, you find an incredible number of variants that have not been discovered worldwide. So that becomes one major reason. But then we give you some examples here, not just doing like large US study, as we all know, we've said this morning, we virtually don't have anything in the global database. But just some very few studies studying genomes of Africans identify some variants that are very, very important for health, for instance. PCSK9, for instance, is one of those that actually is associated, with low density level cholesterol, and that is really has been a target. I'll give you a second one, Xeron B3 is another, variant identified in African population associated with diabetes mellitus, and this really has been also moved forward toward, developing a drug. But then importantly, as we said, you need to study this and take into account the ecology. Variations we're talking about, there are many things, epigenetics could be very important. But what for me is important and then to know is the fact that, we're all doing whole genome sequencing and studying variations across the globe. But I think it is clear that when it comes to, if you look at the history of human migration, just very little group move out of Africa to the rest of the world. So which means that virtually all the diversity has remained in Africa, and it's very important to study the African diversity, and then therefore human-centered atlas will be very important to address some of those things. So this is another case where we talk about kidney disease. APOL1, we all know how it's associated with kidney disease, but it's driven by trypanosomiasis infections, just like malaria and sickle cell. So if you want to study the African genome, so we need to study in the context of our disease on the context of Africa. The context matters, the environment matters, those infections that are circulating in the country, on the continent matters. So it's very important that we actually take that into account. And there's therefore very important to do that in the continent, where we can actually bring all of that together. So you can see here different African populations. We have different groups. Positive selection due to local adaptation is very frequent. We see all of these changes across the genomes on the country, depending on the populations on where you are. So therefore, learning from COVID and learning from what we've done before, we brought in this new platform that we call the Genomic Centers of Excellence in Africa. And that really just describes how we're describing here how we should study human genomics in Africa. And it is based on what I call here a hub and spoke model, where we basically identify hubs of excellence that we have to build, developing hubs and make them hubs themselves. So, and this basically is a network of centers of excellence in Africa that driven, owned, and led by African scientists, is with shared goals, advancing discoveries, and African population and ensuring health security for Africa and the globe. So what makes this very different is the fact that we want something that is agile. We also want something that can be equitable, coordinated, and also build a network across the continent. But most importantly is the fact that it is translational in nature. If you saw the way we started with the infectious disease, we're very translational. We want a network that is translational, finding solution, but not describing, what we're saying. And one of the core is actually a mandate of this network is what? Access and equity. When we talk about access and equity, we're talking about using genomics for equity, but also ensuring that the solutions, that we create access to the solution that we create, we have access to solution that we're creating. So, and then you can see the 4 pillars there. But then this is really the operating principle. Owned, coordinated, and led by Africans, but then the data will be available for people to use. But then through a process where you reach out to the investigators and then you can work in a very collaborative way. It provides support for the translational genomic to equitable mechanism on the continent. It also provides an application for result, for benefit of a community. When I talk about community, it's not just Africa, but actually it's, through the world. So, and we're building an ecosystem, and the value proposition here, is something that is very different. There are many genomic platform networks on the continent, but none, is basically using this approach to address problems. So, in the future, we believe it is very possible for us to basically the workforce development is something that we prioritize, but also making sure that as we are translational, we want to encourage local manufacturing of drugs and vaccines and diagnostics, because that's really where the bottom line is. We can use that with APOL1, as I described to you, to prevent kidney disease, finding cure for HIV, I think delivering a cure for sickle cell, we understand what CRISPR is doing now, but I think we can actually bring it down to a level where it's acceptable. But I think accelerating things like precision oncology for African population, these are things that are very important. We've really made some progress in there. We've been able to register these organizations in the US through the FNIH, and then also have a registration in Africa. And basically we've had a coordinating center, and then the coordinating center is basically just going to be managing the affairs. We have commitment from funders already. So I can say that we also registered organization in Rwanda for the Africa arm. And we do have some funding from some organizations, and then we have orders on the queue. So hopefully in the next few months, you'll hear some announcement. So the goal is actually to, the big picture is to do 3 million African genomes, but we're starting a pilot phase with 50,000 African genomes done in Africa between now and the next year. And that has started, actually. So, and then we believe that you're going to use it for impact, but also ensure that we want to use that as a way to change the narrative. So, and these are, if you want to say, we have a data sharing and data access plan that we've released. We've conducted, socialize this idea. The first time that we discussed with the African community, we had over 1,200 participants from 50 countries in Africa. But then I think the goal is now we have had the pharmaceutical industry or the farmers, they've actually formed an association, and then they're basically working with us in order to drive this project together with a full sense of equity. So imagine a world where we are doing this whole project, and then you have single cell, sequencing data laid over these novel sequences that have not been explored. So this basically just give you, my last slide, to give you some kind of timeline on where we've gone so far. But I just want to say that, these are those really working on this project. So, and then Francis Collins has really been a major supporter as we're doing this work. So I think I really want to end by thanking my team for doing all the work that they're doing, and acknowledge those that are supporting our work. Thank you.
I'm pretty sure that there are lots of observations, comments, and questions for Dr. Happi, but we need to move on. We need to move on because we are running out of time. Our next speaker is Youssef Idaghdour. He is a professor of biology at New York University in Abu Dhabi. He aims to understand how genomes and environment interact at various levels. Today, he's going to talk to us about role of interethnic variation in host response to falciparum malaria. Youssef.
Thank you, Prata. So I think the last talk kind of highlighted the importance of environmental diversity and cultural diversity in Africa. So I'm going to try to really try to convince you that this is something important. It's something that we've been studying in my lab in the last 7 years. So specifically, my lab is interested in understanding the links between genetic variation and phenotypic variation. but more importantly, trying to integrate how we can think about the exposome, generally speaking. So the idea is to look at the environment, lifestyle, infection, and geography, and specifically look at the black box between DNA and phenotypes. We know it's a long way to go from DNA to a phenotype, and this is where the environment is extremely important. So in a quantitative genetic framework, we like to really think about phenotypes and endophenotypes as a product of genetic environmental factors, but also as gene-environment interactions are extremely important in this context. So my lab have been studying these questions in the context of malaria. I don't need to convince you that malaria is a major global health problem. Still, it's not an epidemic. Every year, over 600,000 people die of malaria, the majority of which are children under the age of 5. There is also a huge loss in terms of productivity, and most of the burden is in sub-Saharan Africa. So malaria is quite complex because of the complexity of the cycle, but I'm going to focus mostly on the blood stage of the infection, where the parasite tries to proliferate and the immune system tries to fight the infection. So we've been trying to think about this again in the context of integrating different types of omics data. The idea here is to really think about what happens during the blood stage infection. You have this fight between the parasite and the immune system, and you have the metabolome and the epigenome of both the host and the parasite on top of genetic variation. On top of all of this, you have environmental factors that can impact these relationships. So my lab have been using a population quantitative genetic approach to understand these relationships. So our focus was on a population in Burkina Faso, looking specifically at 3 ethnic groups, the Guan, the Musi, and the Fulani. And the reason for that, there have been a lot of observations from the epidemiology field that there are some very, very interesting differences between these ethnic groups in terms of their susceptibility to or resistance to malaria infection. So the first ethnic group that have been the focus of our work is the Guan ethnic group, and we really struggled to properly come up with a design that will help us gain statistical power for our analysis. It was instrumental for us to try to really kind of get prospective data or matched data. So we've been lucky in Burkina Faso that malaria is seasonal. So when we sample children during the dry season, we have an opportunity to sample them over time. So this gave us an opportunity again to collect paired or matched data. So we did manage to assemble a small cohort of about 150 children. Again, we sample all of them during the dry season. At the end of the dry season, we make sure that the children are not infected with using microscopy, and later on confirmed with qPCR. And then we follow these children over time. At the beginning of the rainy season, that's where malaria transmission starts. And again, because we do weekly or bi-weekly follow-up of these children, we catch the stage that we call asymptomatic parasitemia, which means that the parasite is detected in the blood of these children, but they don't have malaria symptoms. And we keep following them till they have symptoms, the typical malaria symptoms. And we sample them again. Then they are given treatment, and we sample them again 3 weeks post-treatment. Some children clear the parasite, and some don't. And those are very interesting phenotypes that we can look at. So it's a lot of field work, as you can imagine. We have a strong collaboration in Burkina Faso with the Center for Malaria Training and Research, the team of Isiaka Sulama. And again, this really involves a lot of like community work and outreach to really make sure that we are integrating the local population in our program. So we also try to really do as much capacity building as we can. So we're helping the team there to build capacity to do like genetic research. As you can imagine, doing functional genomics is way more challenging. And we've been very lucky to actually set up a small lab there to be able to do single cell work. As you can imagine, it's very, very challenging to do some of these work. This is one day where we couldn't make it to a village where the study participants have been waiting for us. These are just the major challenges that we face in working with these remote populations. You can also imagine that we don't have access to labs everywhere we go. This is a school that was converted to our recruitment site. And these populations are really facing very, very harsh conditions. As you can see, these are conditions where malaria transmission is very, very high in these populations. Now, I want to really go back and focus on very specific ethnic group, the Fulani. This is a group of about 30 million individuals. It spans from West Africa all the way to East Africa. And it's been very, very intriguing for the community because it was reported that this ethnic group tends to be relatively less susceptible to malaria. And there's been several studies that really try to explain why this ethnic group is less susceptible to malaria. I want to highlight specifically the observation that they have low gametocytes in their blood, which actually reduce chances for transmission. But there have been studies that also looked at immune cells and to look at differences in some of these populations. So again, as you can imagine, while working with a pediatric population, it's very, very challenging to obtain large amounts of biological samples. So we developed methods to be able to allow us to really obtain as much information as we can from like relatively small volumes of blood. The idea here is to really extract DNA, RNA, serum, whole blood, and importantly, isolate PBMCs for single cell work. From the infected samples, we try also to capture functional variation from both the host and parasite. And the idea is to be able to integrate all these types of data, as I'm going to show you in a few slides. So a few years ago, we started by looking at the Guan ethnic group, and I won't have time to go into the details, but we used the multi-omics integrative analysis where we looked at global untargeted metabolomic data combined with transcriptomic data, and we showed a very, very interesting observation, which is that these children, when they are infected, their steroidogenesis, those are indigenous steroids are being produced in the blood of these children. So this actually was shown the first time in the context of cancer by actually Sarah Teichmann's group about 10 years ago in the context of cancer, where inside the microenvironment of the tumor cells, T cells specifically, trigger like steroidogenesis. And steroids are very, very potent anti-inflammatory molecules. So as you can imagine, this will have an impact on the immune response of these individuals. But what I want to draw really your attention to is the observation that we only seen this in the Guan ethnic group. So we did exactly the same study. We looked at global untargeted metabolomic in the Fulani ethnic group, and there we see actually the opposite effect. And you're talking about two populations in the same country when children, and again, children, where we try to really control for all the other variables, you have a very, very different response in terms of the response to infection. As you can imagine, the susceptible ethnic group, the Guan, if they have this activation of their steroids, that will dampen their immune system. And we've shown this as well using functional assays in the lab. Now, we don't understand the reason why. We're currently investigating why the Fulani ethnic group, when these children are infected, they're not triggering their steroidogenesis. Another study that we published recently is looking specifically at the impact of ethnicity with a focus on diet specifically. So in this study, we focused on the Fulani and the Musi ethnic groups. They are actually sympatric. They live more or less in the same villages. And the main difference between the two ethnic groups is diet. And so we've been very interested in diet specifically. And again, diet can be extremely important in terms of its impact on immune function. And so because our study design is fully matched, so we control for differences during infection and after infection. Like we can look at the effect of ethnicity and diet simultaneously. As you can see in the PCA here, you can clearly see that, so we have four groups. We have the Musi before and during infection, and we have the Fulani before and during infection. I was very surprised to actually see that the impact of ethnicity is way stronger than the impact of infection on the lipidome of these individuals. So I actually should have mentioned that this is looking specifically at lipids in the serum of these individuals. So this is complex lipid profiling, where we have about over 900 complex lipids. It's expected to some extent that diet would have a very strong impact on the lipidome of these children, but infection also have a very, very strong effect on these lipids. But again, in this very specific case, we see this very strong effect of diet as opposed to infection. In particular, we identified the lipid molecule, which comes specifically from plants. It's called LPC15. It's shown here in this heat map. And in this case, as opposed to the first observation that I told you about the steroid molecules, here you have a lipid molecule that has a very significant impact on the parasite itself. This is actually a molecule that blocks gametocytogenesis, which is something that have been already reported in the Fulani. So we think that we really made a link between a very specific lipid molecule in the diet of the Fulani and their ability to actually have lower gametocytemia, which limits transmission of the disease. So more recently, we, as I mentioned earlier, we collected PBMC samples, and we tried to look specifically at single-cell data comparing the Musi and the Fulani. For this very specific study, unfortunately, we didn't have fully matched or paired data. This is more of a cross-sectional analysis, looking at the difference between the Fulani and the Musi. And we have about 126 single-cell profiles using the 10X platform. And this work was conducted by Tala and Jacob. So I won't have time to go into the analysis, but the idea was to really look at the cell composition between the two ethnic groups and specifically looking at the impact of infection as well. So as you can see here, this is a UMAP map to the reference. We compare the Musi and the Fulani. In general, there are no major differences in cell proportions. CD14 memory T cells and gamma delta are some of the cell types that show differences, but it's not like something that we see across the board. Now, what's important is to, again, because here we can look at the differences between the Musi and the Fulani before infection and during infection. And when we do this, we saw a very, very significant impact of ethnicity on the single-cell transcriptomes. So when we look at the pseudo-bulk data, you can clearly see from the PCA that the profiles, basically the children, cluster by ethnicity, not by infection status. So this is similar to what I showed you with the metabolomics data. The two most prominent cell types where we see very strong impact of ethnicity are monocytes. It's expected because it's also the cell type where we have a lot of power to detect differences due to ethnicity or to malaria infection. So you can see here CD14 and CD16. This is just to give you an idea about those differences, which are really massive. So whether we look at the infected or non-infected state, we see a very, very strong differences in terms of the gene expression profiles of these children. As you can imagine, this would have an impact on how these children respond to infection. We expanded this analysis to look at EQTLs specifically. Of course, we are kind of underpowered to do this, so we use a dominant model. We are very stringent in the way we do this. We look only at cis effects. We detected over 800 cis EQTLs. But importantly, when we look at the interaction effects, which is a SNP by ethnicity effect, we've seen a very interesting observation, which is the fact that there are actually some effects, not many, but we have effects that are actually show a very specific ethnicity regulatory impacts. So in CD36, which is a very important gene that has already been involved in the pathogenesis of malaria, you can clearly see that the Fulani in red, they show differences only when they are carrying the minor allele. Again, this is a dominant model because we want to gain power, so we are only comparing the homozygote reference to the individuals or children carrying the minor allele. So again, these are very intriguing. We don't understand exactly what's driving these effects, but clearly we still have a lot of work to do in terms of functional validation and looking at the impact of these regulatory effects. So just a quick word on the current work that we are currently doing as part of the African Ancestry Immune Cell Atlas in Morocco. This is a project that is funded by CZI with colleagues Sarah Tishkov and others. So the role of my lab specifically is to do this in Morocco and looking specifically at urban and rural populations in Morocco. We are working on the Amazigh, or the Berber people of Morocco. And we just finished the data collection and sample collection, and we are starting experiments in the lab to generate both ATAC-seq and single-cell RNA-seq data. So with that, I would like to thank the study participants, children and their families in Burkina Faso, and members of my lab who did the work. Thank you.
We do have time for a couple of questions. Shyam?
Amazing work, Youssef. I'm really intrigued by how you thought about diet. Often, it would be hard to imagine it has such a strong effect, but how did you zoom in on that?
It was not by design. It just happened that the two ethnic groups that we are working on, so we use self-reported ethnicity, but we collected a lot of information. And diet turned out the single major difference. I mean, there are genetic differences, but diet is the single most important factor that makes a difference between the two ethnic groups. So when we did lipidomics, this was confirmed. And this is, I think this is really important in the context of what we do in most of the presentations that have been presented, talking about the environment in general. Here we are talking at the microenvironment where these immune cells find themselves, which is something that we often overlook. So these are molecules that are extremely potent. The story that I talked about, which is the steroids, that's something that happens during infection, and that will have a very, very strong impact on the cell state of these children.
So does that suggest dietary interventions?
Absolutely, I think that's what we think. Yes.
Yeah, it's very interesting work. Really enjoyed it. My question, I'm sure you have looked into this. What is the distribution pattern of the Duffy trait in these two ethnic populations, and whether there is an interaction between the Duffy trait and some other GWAS you are seeing, which is responsible for the phenotype you are observing?
Yes, so there are definitely genetic effects. I mean, we did control for some major genetic variants, like, for example, the hemoglobin mutations, for example. Because our design is fully matched, and we're interested to look at the differences before infection and during infection, we do control for any genetic differences, because we're looking at the same individuals. When we compare different ethnic groups, it's something that we have to account for. It's not simple. We can use population genetics to control for ethnicity as a whole, but I think the analysis has to be done gene by gene sometimes to look for these differences. But I think this is one challenge that we face, is how we can integrate all these types of information to be able to interpret the results. But yes.
Thank you. Really enjoyed the study design. It was very nice. Our last speaker is Yukinori Okada. He's from the Department of Statistical Genetics, University of Osaka. He's going to talk to us about statistical genetics of individuals, populations at single-cell resolution.
Okay, hello everyone, this is Yukinori Okada from Japan. Thank you so much for giving me the opportunity. So I'm going to talk about statistical genetics in population, individual, and single-cell resolution. So let's think together how we can connect ancestral diversity and cellular biology. So genetics. Human genetics and genome or epigenome is a blueprint of humans. If you can fully understand the meaning of this blueprint, we can understand diversity, difference, and reconstruct individual, but we are still at the tip of the iceberg, and then we wanna go to the bottom of the iceberg. And we are making so many tons of resources, but such resources can be classified in several layers, so population and individual single-cell scales. Population means something we can only get population scale, disease, epidemiology, and individual data, and also we can also get much finer information of single cell within single event. And definitely human genetics is a very nice tool to connect such layers. And genetics, population, ancestral diversity, but single cell may not be so much ancestral diversity. So in other words, human genetics is a very nice tool to connect such population diversity in cellular biology. For example, let me show you some example how we connect each two different layers. So linkage disaggregation score regression from right to left, and single cell disease regulation score. So LDSC is very nice to connect these population and single cell, but their concept is different in terms of directional effects of projections. Okay, let me show you an example. So the LDSC from cells to projection in GWAS result. When we project, we found the Graves' disease in relation to regulatory T cell. Well, when we project these GWAS into single cells, when we project COVID-19 GWAS, we found the causal role monocyte. The point is, such cellular biology reflects the causal roles of disease. Okay, but GWAS is diverse across population. Why not we consider such serotype enrichment in difference in populations? So we focus on some specific, population-specific clinical subtype of COPD, right? There are two types of COPD with chronic bronchitis, alveolar destruction. The destruction position is different, and weight loss and fat. The point is, the majority of these subtypes is different across populations. So let's describe this mystery. So we constructed the population-specific progenicity across respiratory and metabolic traits. So we consider genetics, GWAS, metabolomics, proteomics in both population, and projected the population-specific and progenicity in terms of molecular pathways. Okay, let me show the result of the genetic correlation first. In Europeans, such disease genetic correlations are positive. But in East Asian, we found negative genetic correlation between such respiratory disease and metabolic disease. It means that such negative genetic correlation, especially in East Asian, reflect the epidemiology. And then we utilized polygenic risk score to say the causality, right? And we found from causal reduction from asthma and COPD to types of diabetes in East Asia. It means that even you are trying to get it and fat, it does not cure your COPD. Okay, we confirmed the ancestral diversity. Why not project them with a single serotype specificity? So we project the GWAS result into single cells, but as we expect, the different populations show different enrichment. For example, asthma enrichment, we found B-cell enrichment more in European rather than East Asian. But in COPD regression, we find more fibroblast enrichment in Japanese rather than European. So by connecting these two layers, this genetic single cell, we can now reflect genetic diversity into single cell resolution. Okay, let's go to the next example, how to project. So individual scale to single cell. So Omics project the single cell, so I want to say X chromosome expression in escape. So again, X chromosome expression, one of the X chromosome in females is inactivated, so HCA, right? But 10 to 20% of them genes on X chromosome escape from HCA. This causes the difference of expression levels across sexes. But such very difficult to observe. So along with the collaboration human through others, either led by Shyam Prabhakar, we can see NAX, which is direct observation of HCA escapes in physiological console. Which cell, which haplotype, which gene is escaping? And again, I would like to say, any biology is finally serotype specific. So we found that escape is more serotype specific in lymphocyte than monocyte. For example, such these two genes on X chromosome, but more escaping from B cell or sometimes in T cell. Yes, it's very nice clear, so we can finally discuss serotype specificity. So how we can understand in terms of medicine? The point is, these can explain the difference of GWAS effect sizes across sexes. For example, we always see X chromosome GWAS effect size, but they are sometimes different across males. For example, female biased GWAS effect size can be explained by considering such HCA escapes. So this is not diversity across population, but diversity across sexes. But anyway, when we fully understand the sex bias, we should definitely consider such phenomena. So yes, projection individual data to single cell provides tons of information. Okay, next, going to the next example. So from population to individual and single cell. So population to individual in endogenous viral integration using population genomes, and individual single cell is a Happi viral status projection of single cells. Okay, it's related to the last talk, so mystery of infection and diseases. So here's a long history of virus infection and immune disease. So this is a paper 30 years before my mentor, Professor Yamamoto Kazuhiko wrote, right? They reported a similar sequence, herpes virus and autoantigen. Yes, but it's a kind of true mystery, right? But after 30 years, we have much more better tools. So we whole sequence human include shallow non-human reads. So when we utilize such resources, we can quantify the endogenous virus. So endogenous virus, this is interesting virus genome integrated in human genome, something like human herpes virus 6b. The interesting point is, integration site of such virus into human chromosome is different across population. This is not wide different. But anyway, we apply these methods. And in this paper, we finally found that the difference of herpes human herpes virus 6, for example, 6A is no difference across healthy counterparts. As in 6B, we found quite a higher enrichment as earlier past autoimmune diseases. In people who have such endogenous herpes virus are high active, and then such single cell enhanced interferon response in such, even within phases. So we detect the population using population to individual and projecting single cell, and finally get serotype specificity. Okay, let's go into the final example of from population, individual, single cell, so somatic mutation status. So we have the last day we have breaker section, and we found that diversity in somatic mutation is still completely unclear. And maybe most of here you're now interested in clonal hematopoiesis. So age-related somatic mutation with abnormal chroma exchanges is now risk factor hematological malignancy, but also wide range of human complex traits. But the landscape, especially ethnic diversity, is strongly unclear. There are two types of clonal hematopoiesis: mosaic chromosome alteration and gene-level mutation of chips. Let me show some example of how they are affecting with the human traits. So this is COVID-19 vaccination. So we show germline and somatic mutation on vaccine immunization. We conducted GBAS of COVID-19 response, and as this GBAS result, we can find the two peaks at chromosome 6 and 14, so MHC region and immunoglobulin regions. But we also found the association of somatic mutation. So we conducted the public UK biobank data and found that the chromosome-wide MHC association study, chromosome 6p and 14q somatic mutation is the highest strong risk on COVID-19 vaccination. It's because the two tops in the association with germline and somatic mutation was coincided. And when we focused on somatic mutation, MSC and Age, we find much more strong effects on vaccine. People who have somatic mutation, the MSC has no seropositivity. And such impact also has a risk on Graves' disease. So anyway, somatic mutation is very heterogeneous, but they have specific impact on such immune phenotypes. And then the reviewer asked, how is the, sex chromosome? Yes. Sex chromosome loss also affects such immune phenotypes. And loss of Y decreased vaccine seropositivity. Yes, so this is the reason why the Y-aged male are less likely to be fully vaccinated. But they also have increased the risk of autoimmune allergic diseases and loss of X's as well. So yes, somatic mutation has strong impact on our human health, but we lack the biology. So to understand the biology of such somatic mutation, we should definitely project them into single-cell resolution. So we have recently constructed single-cell deconvolution of clonal hematopoiesis. Okay, let's detect the people who have somatic mutations in population genetics, and using them as a prior information, let's project somatic mutation into single-cell resolution. And we found that there's, finally, serotype specific. For example, the people who just have somatic mutation only in monocytes. But there's different story across serotype specificity. So, for example, these people who have somatic mutation only accumulated B cells, because they are B cell clones, they are BCR is through chromosome. But somatic mutation chromosome B cell, their antigen, when we reconstruct the autobodies, they do not recognize anything. So they keep constructing meaningless IgG. This is the reason why they burn out for infection. But also there is a difference for the loss of Y, which happens when people lose loss of Y. So we found that loss of Y accumulating stimulate specifically marrow cell enrichment. And when the people or cells losing the LOY, they upregulate T cell pathway changes, and then finally T cell fraction or mirrored fraction changes. So LOY may affect immune response via T cell composition changes. But again, the ancestral landscape of how LOY is acting is still completely unknown. And we then looked for the other T cell, because everyone likes T cell, right? And we finally found that neuromediated optical, and it's a kind of autoantigen positive multiple sclerosis. We found they are stronger accumulated in T cell, 21q loss, especially CD4, and the cell which are losing 21q are going to be interferon dysregulation. So, but I want to say that somatic mutation comes with many combinations of somatic mutation chromosomes, serotypicity, diversity, and environment. But we are only in the part of the combination. We should definitely expand the catalog to understand the full landscape of such very mysteries. Okay, again. Anyway, human genetics, germline, and both somatic is very nice to connect ancestral diversity and cell biology. We should definitely keep expanding these catalogs. I would like to thank all the collaborators, especially once I do and go to wonderful lab members. And also, yes, we have just brain biobank at Japan status. Thank you for listening.
Thank you, Yuki, for being on time. We do have time for a couple of questions, if there are. Yes.
Would be great if you could go a bit more in detail in your method on the slide after you had compared the LCDRS with SCDRS. What does your method introduce?
Yeah, we contrast SCDRS, yeah. And LDS previous LDC was developed, and then SCDRS, it's not our method, but here comes the method. They're projecting the tons of GWAS data into each single cell. Because cell number is now increasing, we can get much finer resolution which cell is related to GWAS result. In our cells, we found the enrichment coefficient GWAS risk on especially monocyte stars. Yeah.
Thank you. Let me take this opportunity to thank all of the speakers of this session. And also, very importantly, let me thank Roche for sponsoring this particular session. Thank you very much. Thank you.